I did my veterinary degree, my degree of veterinary science in Berlin in Germany. That's also where I'm from and where I was born. And after I finished my veterinary degree, facing the wide world, being very scared of what's out there, and if I will ever find a job that pays me to buy food, I decided to first work unpaid, because that's what you do back then. Not anymore, don't do it. But I had a dream to go to Australia because I heard about all those weird and wonderful creatures that live there. And so I wanted to check it out after my graduation. So I went to the University of Melbourne to do a research internship on, well, mostly on the creatures, the very furry creatures in the middle, the eastern grey kangaroos. The other ones, ignore them for now, they come a little later. So what I did, I joined the team of the wonderful Dr. Jasmine Hufschmidt at the University of Melbourne, and I did a project on the gut parasites of kangaroos. And it actually turns out they have a lot of them. There's even a hypothesis that they help with digestion and they are slightly symbiotic, but that is not proven yet, but I find it very exciting. Anyway, so I worked with those creatures quite a bit, got to know the Australian culture, mentality, got to really love the country, its nature and its people. And I got the opportunity of something I really always dreamed of, to do a lot of, basically research assistance. And so I assisted multiple PhD students in their field work, and that included a whole range of animals. We went mist netting for birds in the forest, almost stepped on tiger snakes. I helped catching platypus. You think they're cute and cuddly, but the males have a spur, and apparently you never get rid of that pain ever again if you get stung. But they only ooze out that venom during mating season, so don't worry. Then we have the flying foxes or macrobats at the very top. So I got to assist at a rehab center for flying foxes in the tropical north of Australia for a while, which was also very cool to get to know those very special creatures. And then last but not least, the dolphins. I guess that's where it all started. I liked marine mammals before, but my first close contact, so I assisted a PhD student in Western Australia in behavioral studies of these animals, which was very incredible. And and then my very first paper came out of the project with the kangaroos on their gut parasites. And if you had your first paper yet, you know what that feels like. It feels very accomplished, still a bit like, how did that even happen? What is that? But I'm very proud of it. So that was a great feeling. Right, so then the next step, I realized that I really like research. I really enjoyed my time in Australia and my time doing research. See, I missed something. At the very top left for you is the logo of the WDA. Some of you might have heard of it, some of you might not. It's the Wildlife Disease Association. And while getting to know or learning what research means and becoming part of a research community there, I also got to know the Wildlife Disease Association. And I'm still a member, and I've been very active for many years now. And I got to experience what it's like to be a member and to be part of, basically, sounds a bit cliche, but of an extended family that all does work in wildlife research and wildlife health research. So that has been a really exciting journey, and it started back then. So I realized I really enjoy research. I really liked it, and I want to have more of that. And mammal research was the thing I knew I wanted to do more. So I went back to Europe. Unfortunately, it's a bit hard to stay in Australia, you will see. But I went back to Europe, and I got the opportunity to do a veterinary doctorate. The system is quite different there, and I definitely don't want to go into detail, but I like to compare to a research master that I did there. So you are only focused on your research, which was great. And I did it on a very particular group of mammals. You've all heard of them, but I'm sure most of you don't know what they have all in common. So we are talking about the non-ruminating, foregut fermenting herbivores. So we have the sloths, we have peccaries, and we have hippos. There are more, wait for it, but this was a group of mammals that I studied there. None of these occur in the wild in Switzerland, unfortunately. It's a beautiful country, but they don't have those. So I did my studies in zoos, mostly. But what I did was I studied the digestive physiology and the methane production of those animals, because the group I worked with was really interested in, everyone knows that cows, ruminants, produce a lot of methane. They're pretty bad for our climate. The methane is a huge climate change promoter, basically, climate gas. And so the question was in the background, why is it that ruminants produce so much methane? What about other mammals that have a slightly, or that have a different digestive tract? Because all these mammals, they have a chambered stomach, so several stomachs as well, but they don't ruminate. So what is the secret? Why do some produce so much methane and others don't? So it was my job to test how much methane these creatures produced, and ask me for details later if you want to know how you measure methane coming out of a hippo. It is fun. So my doctorate went for 2 years, and the second part of the year was the most exciting one. And probably at that point, I had the most exciting year of my life, because I got a scholarship to go to Australia again, which I wanted to do. So it was amazing. And I got to work with kangaroos, which are the missing link basically in that group of mammals, because kangaroos also have a chambered stomach, but they do not ruminate. Or do they? No, they very likely don't. So they're part of this group. So we wanted to find out how much methane do these creatures produce, and how does their digestive physiology work in that regard. Yeah, it was an amazing time. As you can see, it was quite far and remote from everything. I stayed at a remote research station called Fowler's Gap in the Australian outback for almost a year, and met the most incredible kangaroos, the most incredible people. So I had a really amazing time there. And this was actually, it is still the research station of UNSW in Sydney. The logo is at the very top right. And so it was a bit of a big collaboration between several universities, this project. And we will get to that university later, because that's where I did my PhD afterwards. But first of all, I know you're all dying to know, so tell us, how much methane do kangaroos produce? I'll tell you. So I think how my supervisor got the funding for this was basically like, we need to do this experiment to have a better argument in case kangaroos produce less methane, that people eat more kangaroo meat. This might not be as much of an issue here in the States. We can push this a little, but it's mostly an Australian thing. But we did find that kangaroos do produce way less than ruminants, like cows, like all the other foregut fermenting mammals. They rank way lower than the ruminants. Maybe not a huge surprise, but no one had really checked it before. So here's a really good argument to eat more kangaroo meat next time you get a chance. I'm vegetarian, I haven't tried it, but I heard it's not too bad, and it's mostly very ecologically friendly, because they're not farmed, they're all shot. So they had a fairly happy life, most of them. Right, so then there was a little side project that I did. This is a very funny one. So I bet most of you have never heard of this term, meriscism in kangaroos. It's a little bit like ruminating, but not quite. So it basically means that you regurgitate your food. You as a human can do it too, don't worry. And then you chew again and you swallow it. But again, it's not the same as rumination, because rumination is it's essential, meriscism not necessarily. And I noticed, especially in the younger kangaroos, that they did that sometimes. And I know the pictures are pretty crap. That's what ended up in the paper. So, the right picture is a juvenile eastern grey kangaroo, and that's just basically regurgitating and spilling the food it just ate in a big rush because all his friends wanted to eat the food as well. So I studied that for a little bit as a side project because that had never really been investigated before. And they do it sometimes. We still don't really know why they do it, probably because they don't chew properly, those lazy things. And we don't know how common that is in the wild. So definitely, if you're still looking for a research project, maybe not here, but I can recommend that. There's still lots to do. All right, let's move on. So I finished up my doctorate, had an amazing time in Australia, went back to Europe again and applied for a PhD because I knew, yes, I'm really interested in research. This is what I want to do. And I love working with mammals, but I want to get into marine mammal research and especially marine mammal health research. That's what I kind of knew was always my thing, but it sometimes takes a while to get there, especially when you are from a very landlocked place called Berlin. Germany does have an ocean, by the way, just saying, but it's not very impressive, but Berlin does not. So there was a bit of time in between. It didn't happen straight away. But I got there finally to do my PhD in Sydney in Australia at the University of New South Wales in Sydney. And that was really a dream come true. True, not true, that as well, but true. Because I got to study the respiratory microbiome of cetaceans, their drivers, and their potential correlation with health. It's a bit one hell of a title because yeah, it's a huge topic and definitely way more than you can find out within one PhD. But you know, when you are young and at the very beginning in your honeymoon period of your PhD, very ambitious. So I studied the respiratory microbiome in humpback whales and in dolphins. And these were the species I had available. And if you've never heard of the potential relevance of respiratory microbiome, you might be like, why? Good question. Don't blame me for that. So I'm sure you have all heard of the gut microbiome, and obviously for decades now, this has been a huge topic that we need to keep our gut microbiome healthy because it helps to keep us healthy. If our microbiome in the gut is not healthy, we probably have digestive issues, we might get mental health issues, there's a whole lot, it's all connected. And it might be the same, maybe a bit less with the respiratory microbiome. And the issue is with cetaceans, especially in the wild, that it is really hard to assess their health because, well, you can tell here, so we can basically the easiest thing is to wait for them to die and then to pick them up and cut them open. But then you can't really tell how they, well, if you ask, how is this dolphin doing? Well, it's dead. So it's not really, like it doesn't tell you how it was doing when it was still alive. So the idea was to investigate the bacteria, the microbiome in the blood of the whales and dolphins to see if that has any correlation with their health. And again, in the literature for humans and for lab rats, mice, there's definitely some evidence for that there's a correlation and a bit of an interaction with the immune system as well. Things like asthma and COPD and other respiratory diseases are definitely linked to the microbiome in some way or another. So as I said, I worked on the humpback whales, but so the humpback whales, there is a massive population of humpback whales off the coast of the east coast of Australia. They travel past the coast every year twice. So they spend their summers in Antarctica. Where else would you want to go for your summer holiday? It's great. And then they travel up the coast to their warmer mating grounds in the north, in the tropical part of Australia. And so it's a huge population by now, about 40,000, one of the very few cetacean populations in the world that has recovered so well, which is incredible. They were reduced down to an estimate of about 200 individuals. So maybe a bit of a bottleneck there, but they're not suffering just yet. Yeah, so it's just amazing. You are just standing on a cliff watching over the ocean and you can see them really close by. So it is definitely an amazing place, not just for research, but also for whale watching. But the issue is, and I will get to that in a moment, when you sample the humpback whales, and and analyze their microbiome in their blow, that's great. And I did get some results from that, but they don't tell you anything. They don't tell you like how old they are. They don't tell you, sometimes that you can't even tell what sex they are. So the information you get from them is a little limited. So that's why I refer to the closest equivalent of the lab rat in cetaceans, there's dolphins under human care. Yeah, so that's why I studied dolphins as well and looked at their microbiome. But yeah, let's have a look at that study first. So obviously my PhD had a few chapters, but this is a highlight. Don't want to bore you with the chapters I'm not that proud of. No, they were all right, but these are definitely the more exciting ones. So I work with SeaWorld in Australia, which is not at all related to the US SeaWorld. Why haven't they not been sued by the US SeaWorld? I have no idea. I guess they're just lucky. Anyway, so the trainers in SeaWorld there were very, yeah, I was very grateful to them. They collected the blow samples, as you can see on that picture, in that way, from 10 dolphins over 8 months, once a week. And then they sent me the samples and I analyzed them. And the idea was to basically investigate how stable are their bacterial communities. Are they stable? Which means, do they stay stable over those 8 months? Do they stay similar? Do they change? Because if they change all the time, it's very unlikely that there's a correlation with their health, because then they're probably just like window shopping and passing by, being breathed in one moment and exhaled the next. So that's why I wanted to know how stable are those communities in there. But I also wanted to know other aspects, like what about factors like age, sex, the pool the dolphins were kept in, what impact had that on the microbiome? So yeah, in the end, I had all the samples, a lot of dolphin breath, frozen dolphin breath, and I analyzed it all, and it actually turned out that all of these factors had an impact on the microbiome of the dolphin. So the age of the dolphin, the sex, the pool they lived in, it all shaped the microbiome, which makes a lot of sense. It's a very dynamic system. But I also found that each individual dolphin also had their individual microbiome, which was fairly stable over this time period, and basically stay true to its dolphin. So there's at least a chance that there's a certain level of symbiosis going on, potentially. And the dolphins, across dolphins, in the comparing each individual or all the individuals with each other, they also shared a certain number of bacteria, which gives us also evidence that it's not all random. Those bacteria that they had in their airways were specific to dolphins and were actually found in cetaceans before. Might sound, if you work with systems that are much easier to be controlled, then you might think like, ugh, what a mess. Yes, that is marine mammal research. So that's the best we can do. So yeah, I was actually I was really happy about this chapter. And again, it definitely gave us a hint that there is stability in those microbial communities, and they might be useful as a health assessment tool. So then I already gave you a teaser. There was another chapter on humpback whales. And to give myself a break, I will let the video speak for itself.
Wait for it. That's UNSW's Dr. Catharina Vendl collecting samples from the blow of Eastern Australian humpback whales.
I would get ready with my long pole in the petri dishes and try to be there at the right time, at the right spot, to extend my pole and my whole body basically over the rail and try to catch that blow when the whale was exhaling.
Tricky but important work to understand how the epic annual migrations that whales undertake affect their health. Each year from around April, thousands of whales leave their feeding grounds in Antarctic waters and travel north along the east coast of Australia until they reach their breeding areas on the Great Barrier Reef. And then in late winter and spring, they return south after 4 months of very little food.
There's just not enough food available north of Antarctica for them. They're big creatures. They need a lot of food, not available. So they live off their energy reserves. They lose up to 50% of their body weight during that time.
Researchers know this from studying aerial photos taken at various stages of the migration. But by collecting the samples from the whales below, they get much better insights into their overall health. That's because the airways of whales, just as with other mammals, contain large communities of bacteria. The first samples were collected early during the whales' migration off the coast of Sydney.
At the beginning of their migration, they had quite rich microbial communities, which means that there was a large variety of different bacteria.
More samples were taken 3 months later in Hervey Bay off Queensland.
The special thing about the sample site is that the whales often approach the boats.
But when they didn't, the scientists used modified drones to hover over the whales, ready to be sprayed. Still, even that posed challenges.
You never know when the whale will exhale. You never know when the whale will come up.
It was worth the effort. The sample results show that months of fasting seriously depleted the whales' bacterial communities, likely weakening their immune system. Ongoing tests will be important to chart the overall health of whale populations. Dr. Vendl says it's the type of fieldwork she will never take for granted.
I'm from Germany originally, from the city of Berlin, and we don't have that. And although I've been living here for like more than 4 years now, but it still amazes me that you can just go out there to the headlands anywhere around Sydney and you see them migrating past. I think that's pretty incredible.
The Australian Academy of Science, because questions need answers.
I'm still amazed they made that video. Anyway.
Wait.
Not again. Okay, so now the question remains, how good is analyzing the below microbiome in terms of assessing the health of a population? Well, it's a very important question, a very good one. I got asked that a lot when I finished my PhD, so I'll try to answer it. So the situation is complicated. And as I mentioned before, as in most research areas, so much more needs to be done. So I could show, or we could show with my colleagues, that the microbiome has a certain level of stability, but is also very specific to the individual. So we would have to learn much more how much variability there is and what the drivers of that variability are. And I only did 16S rRNA sequencing, which only shows you a tiny window of that microbiome. Often doesn't even tell you the species of the bacteria. So a whole shotgun sequencing would really help to look into that more. So definitely more work there. And I will get to that towards the end, because in the Carmichael Lab, there's some ongoing work on dolphin microbiome as well. So yeah, I'm glad that I'm not done with that topic. All right, so then next step in my career journey, I had finished my PhD, the pandemic started hitting, and I got picked up from the street and got a postdoc position. Whew, that was close. So I started my postdoc in a different research group, but at the same university. And the research group, well, the topic was very different. It was a research group that worked in mostly meta research only. So for those of you who are a bit like, what is that? Sounds complicated. It is, I can tell you. So basically, I will get to that a bit more, but what it basically means is that you use the data that has been published before, mesh it all up, use very sophisticated stats, and get amazing results out. And that's what we did on a very particular topic, which I got to love, not for its health impacts, but for the topic itself. So I worked on PFAS. I'm sure some of you have heard of it, some of you might not. And those of you who haven't, I'm sure you won't stay innocent forever, because it's all over the media. It's becoming more and more the topic of the I don't know, century probably. So the PFAS stands for well, you can read it because I never get it right. And they're nicknamed forever chemicals. They are a very large group of man-made chemicals, more than 5,000 different components, and they are extremely useful. So it's easy to, well, to say like, oh, why did we even invent those? Horrible. But to be fair, that would just be one very ignorant side of the scheme, because they have been incredibly useful and have basically changed our lives. So what you can see here, all those different products, they, well, some of them essentially depend on PFAS, others have it as a nice add-on. But where, for example, the most famous representative of PFAS, or where you use PFAS, is Teflon. So Teflon, the non-stick frying pans, they have one of the PFAS component as their main element to make sure that your steak or whatever doesn't stick to the frying pan. Because what PFAS does, and that makes it so amazing, one of the things, it prevents grease and water sticking to surfaces. And when you're going out hiking and you're wearing a great $300 outdoor jacket, you do not want to get like soil and mud stains on it. That's why it's impregnated with PFAS. Amazing. And what is crazy is that there is still, you can correct me if I'm wrong, but I'm not aware, there's still no viable alternative for it. Some companies, which are more, try to be more green, they use bee wax now, which is such a pain. Anyway, I'm getting into too much detail. You can tell I've spent a lot of time on this. But there are literally still no really viable alternatives. They are better for the environment, but they don't have all the other amazing properties that PFAS has. But enough about praising PFAS, because it's also evil, right? It's like not black and white, it's very gray. So forever chemicals, because they do not degrade in the environment. They have such strong, so their secret are the carbon and fluoride, or carbon and fluor bonds, and they're extremely strong, one of the strongest bonds in chemistry. And that means, yeah, they do not degrade. Basically, every living creature on this planet has some level of PFAS, some level of concentration in mostly your blood. You keep it for about 4 years, or like you slowly eliminate it over about 4 years, but you always ingest new of that stuff. So you basically can't escape it. And in the environment, like it ends up somewhere in the environment, including the oceans. It just stays because it has nowhere else to go.
Do you like prawns? They're pretty delicious, aren't they? The only issue with prawns and other seafood is that they come with a cocktail of man-made chemicals, including PFAS. PFAS are used in many different products, like non-sticky cookingware and firefighting foams. To date, PFAS is almost everywhere in our environment, and seafood is one of the main sources to get exposed to it. High doses can increase the risk for many different illnesses like diabetes and breast cancer. But fear not, we found an easy fix for you. In our meta-analysis, we analyzed the outcome of 10 studies that cooked seafood in different ways to reduce PFAS. Cooking time and volume of cooking medium, like water or oil, had the largest impact. For example, if you boil 200 grams of prawns in 2 liters of water for around 11 minutes, you will reduce the PFAS content by 75%. You can't eliminate PFAS completely, but it's a pretty good start. Enjoy your prawns almost PFAS-free.
So just as a reminder, 200 grams of prawns cooked in 9 liters. Okay, it's a lot, but you know, it's for your health. Of water for about 11 minutes, PFAS reduction of 75%. So yeah, this is really just the tip of the iceberg of what we found. We had 10 studies with very different types of cooking, boiling, steaming, frying, cooking with oil, cooking with water, cooking for a short time, cooking for a long time. And again, put it all in a pot, or like applied very fancy stats to it. And then we got the results that the cooking time and the volume of cooking liquid, doesn't matter if it's oil or water, had the biggest impact. So next time you prepare your seafood, make sure you use as much water and as much oil, or one of the two ideally, gross otherwise, as much water or as much oil as possible, and you cook it as long as possible. Don't cook it until it's all black. Other health implications, but you know, it's about compromise. All right, then another highlight of my work and my postdoc, it's fresh from the press. So yeah, I'm very happy that it just came out. This is also a piece of meta-synthesis, but in this case, not a meta-analysis, but called a systematic evidence map. If you haven't heard of it, it is basically creating a database of all the literature that is out there on a specific topic. In this case, about PFAS in wildlife and what research has been done, mostly about distribution and abundance of PFAS in wildlife, but also health consequences, impact on the immune system and other aspects. And yeah, so this paper collated all the data, applied some stats to it as well, and presents it as basically showing what has been studied to a large amount, and where are the gaps, like what needs to be studied more to make it really a comprehensive body of data. I have a few representatives down here. So we actually found that the common carp right here was most studied, and the majority of papers studied carp, and second ranked the polar bear, because it actually turned out the polar bear is not only a very popular face for climate change, but also for PFAS. They're at the very top of the food chain. They live comparably long lives. That means they bioaccumulate quite a large amount of PFAS over their lives, unfortunately. If you're interested in that field, check it out. I can recommend it. All right, then I want to give you a very brief overview of what I'm doing now. So as I was introduced, I'm I started in May, still feel a bit new, the research veterinarian and veterinarian of the Alabama Marine Mammal Stranding Network, within the Carmichael Lab. And that comes with a whole bunch of tasks, which I actually very much like about this job. One of the aspects I really like about it, it comes with quite a variability of tasks. So it definitely never gets boring. So that's pretty cool. No one wants to be bored. But first of all, some of you might wonder, what is she doing here? Like, how did she get here? This is random. Yes, it is very random, I can tell you. So this is how it all started. I'm sure many of you will remember the really lovely Jenny Bloodgood. She was the previous research veterinarian here, and I met her at a conference in 2022 in Madison, Wisconsin. We got along well. She's obviously super nice, and I got to visit her here on the island. And really, the excuse was that I wanted to interview her for a podcast I've been doing for a while now. So I'm really into science communication, and I have been doing podcasting for probably 7 years now. And this is a newer podcast project, which I've been doing for about a year now. And I will show you what it looks like. But basically, I came down here to the island and did an interview with Jenny and asked her all the questions about her work, because I thought her work is really cool, working on such a beautiful island. And that's how it started. And that's how I got to know the people and how I got to know the place, and even learned about that there's a thing called Dauphin Island Sea Lab. Amazing. So if you're interested in podcasts and wildlife health by any chance, we also talk about fish sometimes. This is the podcast. It's called Wildlife Health Talks. It's a podcast basically hosted by the Wildlife Disease Association, but I run it and I do the interviews, and it has been really fun. And I've been getting to know a lot of amazing people, including Jenny, through that work. It has been really fun. All right, but so this is how I got into the whole thing. Jenny got a new job, and then I got her job. And what do I do here now? So as I said, there's quite a range of things, which is exciting. So I'm the instructor for the Marine Mammal Health and Stranding Response course in summer here as part of the Sea Lab summer program. So I've done it already last year. It has been great fun. I've really enjoyed it. So this is coming up again this year. Then I'm also, as I mentioned, the veterinarian for the Stranding Network. So most of you probably know, who had a glimpse through the window in our mammal building, most of the animals are dead that we are getting. So what we do is we do necropsy and then we investigate the cause of death. So we are basically dolphin detectives, and which is also very exciting. And then I do research. So I started a few research projects around marine mammal or cetacean health. So, and at the end, I'm doing so well in time, I'm amazed. So just a little case for you. If you're grossed out by necropsies, maybe leave now. You still have time. Okay, so this is the dolphin Medi. Medi's was a pretty dolphin, and then he died. So the cool thing was also he was a necropsy. We did a necropsy on him as part of the Marine Mammal Health course. So the students got to experience this as well. And there is quite a bit of a twist coming, so they really enjoyed it. So I hope you enjoy it too. Well, it's sad, but it's also very cool. Okay, so Medi was a subadult bottlenose dolphin. He was reported dead in Fort Morgan, and he stranded in January last year, so about a year ago. And then 6 months later, we froze him to use him for teaching purposes, and 6 months later, we did the necropsy on him. First, looks gross, but that's what they look like when you cut them open, right? So first Medi looked really healthy, everything pretty good, very healthy, strong male. So I was like, well, what happened to him? Then we got into it a bit more, and then we found that something also as an explanation. So this is what we call the plug. This is the you can't see it, but there's the tongue to the very left. Then we have the trachea, and we have the lungs, and in the very middle there is the heart. The heart is very red, and it's not meant to be that red usually, not that dark red at least, because that arrow is pointing at a huge blood clot, which should not be there, because if you have such a big blood clot, you lost a lot of blood, not good. So we found that, well, actually Mackenzie found it. She was working on that, and we were like, we wonder where that's coming from. That's really interesting. And then we had a closer look at the heart. So the top part is the heart, very pretty dolphin heart. And at the bottom is that blood clot after it was kind of like peeled off the heart. So the heart on one side was basically surrounded by this massive blood clot. So where did it come from? What happened there? And I have to say, it is really important to know that you do necropsies. And in most cases, when you look at the dolphin in the gross necropsy, you do not find what they die of. You can, but in a lot of cases you don't, which can be really frustrating. But Medi did not disappoint. So when we had a closer look, we actually found a spot where I think it was the left ventricle, not sure, one of the ventricles was perforated. That's what's marked here. And what happened there was something perforated that poor dolphin's heart, and it was that. He basically got stabbed by, you might guess it, a stingray. So death by stingray. So yeah, that sometimes takes a while to sink in. Anyway, so Medi died of a peracute perforation he died of a perforation of one of the ventricles, basically bled out, and the heart would have eventually stopped, not having enough blood to be pumped around the body. So case solved in this case. And this is really just a brief wrap up. I'm working on other projects. I mentioned before microbiome, and keep pointing at Joe because he's working, a lot of you I'm sure know that he's working on skin microbiome in our group as well. So there are a few projects going on. But I started taking over some of the work from a previous RU student, and I'm working on the necrobiome, which I haven't done before. I find it very exciting. And it's basically to investigate how does the microbiome on the skin or of the organs change once the dolphin is dead. And that will hopefully tell us a bit more about how does decomposition actually work? Like we all know that they decompose, but how does it work? Like where do those bacteria come from? And can they give us a hint about how long ago the dolphin died, depending on what the composition looks like? Many, many questions, no answers yet, but hopefully we will get to that a bit in the near future. And this is a lovely Venn diagram that I made myself. I just want to tell you that I presented to you a lot of different things, a colorful mix of things. And I know that a lot of researchers and academics are like that. They're like, oh, this is interesting and that is interesting and that. But you also have to be a bit focused, right? Otherwise, yeah, you only have so much time. So what I'm really interested in these days is investigating the drivers of marine mammal health. So what is it, or especially cetacean health? So what is it that keeps them healthy? What is it that contributes to the immune system potentially being weakened? What is it that makes them sick? And there are obviously so many different components. There are contaminants like PFAS they are exposed to. There's the microbiome, which they probably have a symbiotic relationship with. So, and these are really just two of the many components that impact the health of cetaceans. And I find it really fascinating. It's a never-ending story. And yeah, and that's what I'm working on and we're working on in our lab. And this is just the diagram to show that if you're not a dolphin fan, that is okay. Not everyone has to be a dolphin fan, but of course, dolphins don't stand alone in the ecosystem. They eat fish. Fishes have, just as an example, fish have PFAS, dolphin eat fish, dolphin accumulates PFAS. We eat fish, we accumulate PFAS. So in the end, it is all connected. Ocean water has PFAS in it as well. So again, just to illustrate that it's all connected, and I'm sure you've all heard of the term One Health. It's a really important one. It has been very trendy, but for a very good reason, because studying cetaceans gives us a little window and a little hint into what we are facing and what drivers are actually impacting our health in the sometimes short term, but definitely in the long term. And that's it. And before I take questions, if you have any, very happy to answer them. Just want to let you know, I'm sure I've met most of you. If I haven't, please come and say hello. I feel like I still haven't met so many people in the Sea Lab, and it's sad. I want to meet everyone, so come and say hello. And yeah, if there's anything you're interested in, what I've worked on, please come and talk to me, and I love to chat about those things. So if there's anything you might want to collaborate in the future, come and talk to me as well. I would love to have that chat. All right, thank you very much. Any questions? Yes, Lee.
I think there's your method from a hippopotamus. I mean, on the edge of my seat,
Right.
Power.
And I really wish I had a picture, but I could not find those pictures and those photographs last night. Anyway, very good question. So it was such a pain, I tell you. So, by the way, we're talking pygmy hippos, because the big ones just would have been, logistics would have been too difficult. So basically what we did with all of the animals, we put them in a box. So like basically a metabolic cage. And it doesn't sound great, but you know, they were all unharmed and they lived happy lives after. So yeah, we basically built a crate made out of wood, which is not ideal because it's not fully sealed, but you can only do so much. So we built a crate, which is a bit like a transport crate you have for hippos. And it was like a long, fairly narrow crate, so the hippo could not move very much in there, but it was only in there for a day. Yeah, so we put it in there, we sealed it, and then it was attached to those measuring devices, which would measure not just methane, but also, yeah, obviously carbon dioxide, oxygen, and all that. And that's how you do it. Easy. You also have to make sure you don't get eaten by the hippo in the process, because, you know, when
Dr. Baker was telling me to sit behind him with a cigarette lighter measuring a flame.
oh, that's actually such a good point. Thank you. Because so thank you, Lee, because we have to get rid of that myth. So I'm so glad you're my guinea pig, because it's really funny. My old supervisor, he's not old, but you know, back then, Dr. Markus Klaus, he's an expert on like nutrition in zoo animals back in Switzerland. And he hated it when people made those fart jokes. He hated it because I think he felt like people were putting him in a box for him. But what actually the situation is, and I don't blame you because everyone thinks that, but what actually the truth is that most of the methane is exhaled through the breath. And that's even the same with ruminants. Usually they burp it out or they just exhale it. So of course they do fart, but it's only very small volume of methane. And the other thing that a lot of people don't know is that methane, you cannot smell it. Most people think, oh, when you fart, you know, then the smell is methane, but it's not. It's actually like a sulfur gas, not a gas. Anyway, so yeah, so methane is not as bad as its reputation. Yeah. And that's also why you have to, yeah, exactly why you have to put them in a cage instead of just measuring what comes out of the end, because that would only be a very small ratio. Any other questions? All right. Sure.
The previous research is that the entirely meta-analysis. We performed analysis to monitor contamination and
Oh, that's a really good question. I don't know who asked that, but thank you. Yes, so there's an ongoing collaboration with Professor David Haller from Texas A&M University. He is a PFAS guy. He has all the very fancy machines, which are very, very expensive. We don't have those here. Well, to be fair, there are people here at the Sea Lab and at South working on that as well. But the Carmichael Lab doesn't have those machines. But we have that collaborator, and we sent him some time ago, we sent him dolphin samples, and that paper actually just came out as well. So we do measure PFAS levels in our animals as well. And we just agreed on continuing the collaboration. So there will be more in the future. So stay tuned.